Hello, and welcome to this episode of my podcast. Today we're going to deep dive into the fascinating world of CRISPR and genetic editing. We'll explore what CRISPR and CRISPR-Cas9 is, how it works, and its potential for treating genetic diseases. We're going to follow that by discussing the different types of genetic alterations and how each of them provides their own benefits to humanity, and their ways of completely destroying it, potentially. We'll also discuss the ethical implications of gene therapy and some reasons why it can get out of hand so quickly. And then lastly, we're going to discuss what the future holds for genetic therapy and what we think we can see from it in the next 20 to 40 years. I am Ryan McCoy, senior and soon-to-be graduate from Platte County High School, and I've made it one of my top priorities to research gene therapy over the course of the last couple months, because I think it's something more people should be aware of. It can change the world, so who wouldn't want to learn more about it? And I also have a guest with me, Jordan Hedrick.
Hello.
Who has also been researching CRISPR recently for his own research topic. So he's going to provide some knowledge on that front. So I think it's pretty important to just start with defining what CRISPR is, and where it came from, how it works, and all that good stuff before we get into the actual topic of genetic editing. So CRISPR was discovered in 2012 by two scientists, Jennifer Doudna and Emmanuel Charpentier. They worked together to discover the combination of CRISPR and Cas9 protein to be able to cut segments of DNA to edit genes. So with that being said, CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats. It's an acronym. It's a system that bacteria uses to defend themselves against viruses. So it's found in your body. So it's not really something that Jennifer Doudna and Emmanuel Charpentier created or made. It's something that they discovered in the body. And scientists have adapted the system of bacteria for genetic editing in human cells over the last 20 years. It's advanced rapidly. So it allows scientists to modify or remove specific genes in the genome.
So you're saying that CRISPR basically takes part of a human's genes, like a specific strand,
Yeah.
and removes it because it's infected with bacteria or viruses?
Yeah, so it consists of two main components, the Cas9 protein and a small RNA molecule. And the small RNA molecule is CRISPR. So it's a cRNA, it's CRISPR
Gotcha.
RNA. So the RNA molecule guides the Cas9 protein to the specific location in the genome where the modification or the deletion should occur, so where that segment gets cut. So once the Cas9 protein is at the target site, it makes a cut in the DNA strand. The cut triggers the cell's natural DNA repair mechanisms, which can be harnessed to introduce specific changes into the genetic code. That was really kind of an in-depth scientific description of it, but there's kind of a simpler way of putting it. So if you think about it this way, it is, you can think of it as CRISPR, the CRISPR RNA molecule being a paper with like a dotted line, from elementary school, like a piece of paper with a dotted line that you're supposed to cut. And then you think of Cas9 as the scissors that are cutting the paper. So the CRISPR RNA molecule guides the Cas9 and shows it where to cut, and then the Cas9 cuts it. And following that, that segment of DNA is just eliminated and replaced with a new, healthier segment of DNA, causing it to completely change the genome in and of itself.
So essentially, you're saying that CRISPR RNA finds the unwanted strand within the genome that has been infected by a disease or bacteria that is overall hurting the human body system, and CRISPR cuts that selected portion out to get rid of that disease.
Yeah, so not necessarily like an infected segment of DNA, but if someone was born with a genetic disease, say like cystic fibrosis or something like that, they'll have a segment of DNA that's causing it, so scientists will target that segment of DNA, and then the CRISPR RNA, which is a guide RNA, will guide the Cas9 to that segment of DNA, and it'll just cut it right clean off, and then it'll get replaced by a healthier segment of DNA, in hopes of potentially eliminating that genetic disease from the embryo. Yeah.
So you talked about how is it normally done at a young age, like when they spot it at birth or something?
Yeah, I'm going to kind of get into that later. There's a couple different types of genetic editing. There's 3 different types to be exact. That's something I'll get into a little bit later on in the podcast. But it kind of depends. You can do it in an embryo of a kid, or you can use CRISPR and gene therapy to try and treat certain diseases that people already have. Yeah, so I mean, there's a couple different types, but I'll be getting into that later in the podcast.
So you're saying that you can get CRISPR done while as an embryo, so like within your mother.
Well, it wouldn't be if it's an embryo. It'd be before the mom starts to develop it, in her stomach. It'd be it'd be 2 gametes, like premature gametes. So premature, so the sperm and the egg,
Gotcha.
Creates the embryo, and then from then they would work to eliminate those harmful genetic diseases before that embryo gets inserted into the woman.
Gotcha.
Yeah.
And like, how successful are all of these? Is it like a surgery?
I mean, it'd be through in vitro fertilization. So that's if you didn't know, that's like almost manual pregnancy.
Mm-hmm.
And so that's how it kind of has to be, because you can't really like genetically edit an embryo that's already with natural pregnancy.
Yeah.
But it's a really new topic and a new thing that in science. So there's not really necessarily a number on it of how successful it is right now, but there's so many examples of how it's been used, whether it be legally or illegally, from some foreign countries and things like that show how powerful it can be in the near future. So.
Like, say you also said that you could do it while you have the disease already.
Yeah.
Is
so
that successful?
Yeah, I'll get into that here in a little bit.
Okay.
That's gene therapy, and it's not necessarily using CRISPR. So I'll talk about that in a little bit and how it's different. But it kind of stems from the same thing in trying to eliminate genetic disorders and conditions that some people have. And that's a lot less successful because obviously the person already has that condition. So it's a lot harder to just eliminate it. But, obviously, that's something that scientists are working really hard to try and master as well. So I'll continue with that later. But I understand that you had a research topic involving CRISPR as well, Jordy. So can you explain a little bit about that topic and why you chose it and how CRISPR is used within your topic of research?
Yeah, my topic was actually played last episode, if you guys want to go check that out. But CRISPR can be used with the kidney growings that I talked about. Or I guess I'll just give you a brief little description right there. But overall, CRISPR helps find and address diseases that scientists haven't been able to discover and understand within kidney problems. For example, we have like polycystic disease and nephrotic syndrome, which these have been determined through the kidney organoids that wouldn't have been able to do without the CRISPR genetic editing.
Yeah, so you're saying that the CRISPR is going in the segments of DNA inside of the kidney, and it's eliminating the harmful diseases. Is that what you're?
Yep.
So, yeah. So basically, kind of the same
Thing as
sort
like
of idea.
genome,
Yeah, exactly.
it's inside of an organ.
It's inside of an organ instead. That's pretty cool.
It's pretty neat to think about.
Yeah.
Like all of the genome.
Yeah, and the fact that CRISPR has the power to do so many different things that you wouldn't even think of. So I'm glad I got someone here that's familiar with the topic. And so I guess now that we've kind of covered what CRISPR is and a little bit of how it's being used medically to the best of our abilities, I'm going to just segue into the broad explanation of gene therapy and how CRISPR is being used within that portion of science. So, like I mentioned earlier, Jennifer Doudna and Emmanuel Charpentier created CRISPR, and they created it they discovered it with the goal and the intent to eliminate genetic diseases such as sickle cell anemia, cystic fibrosis, any genetic disease. I personally feel like that was their honest intent, but there's actually a lot of speculation out there that their intentions were different than that. And I'm going to go into that a little bit further and what that pertains. But throughout the last 10 years or so, Jennifer Doudna has made it very clear and continues to echo that although genetic alteration is extremely powerful, the only true use of it should be to cure and fight off genetic diseases that were said to be incurable, because it's a technology that is powerful enough to do something like that. And so despite those the outside concerns that some people think they discovered it for, and some people think they were researching, we're going to talk about that later, but the potential of genetic editing is extremely immense, and we may see a world in the next 20 to 40 years where genetic disorders are completely eliminated and chronic illnesses are treated with one single therapy.
Awesome.
Yeah, and so we may see the development of personalized medicine tailored to the unique genetic makeup of each patient, so it completely eliminates every potential genetic disease that they could have from each parent, passed hereditarily, which would be just completely revolutionary for all of biomedical science, and it would completely change the face of the world and the health of the world. So that's pretty cool to even just think about how powerful the CRISPR-Cas9 technology that Doudna and Charpentier created or discovered really is. So now that we've kind of covered everything about CRISPR-Cas9 technology, and now that everybody has a somewhat of an understanding of how it works, I kind of want to get into the different types of genetic alteration. And so there's 3 different subcategories, but basically genetic alteration is the overarching term used to describe all the different types of gene editing. CRISPR-Cas9 technology, discovered by Doudna and Charpentier, makes it possible to address flaws, repair mutations, cure genetic diseases, and maybe even splice DNA from one animal into the other. So when you think about something like that, you can think of a million different procedures and outcomes that could happen from that. But overall, when you're talking about genetic alteration, there's 3 subcategories, and so it's vital to know the difference between the 3 categories. So I'm going to kind of go and explain each category, how it works, and the specifics of each one of those. And so these 3 categories include gene therapy, hereditable genome editing, and genetic enhancements. So these 3 categories are very important to understand when you're talking about genetic alteration, because one is already existing, and it's getting used on a day-to-day basis, and one is just now being discovered and just now starting to get used, and one is kind of what you could see in the future as genetic editing enhances. And so I'm going to start with a pre-existing genetic alteration category, which is gene therapy. So we talked a little bit about that earlier. I said I was going to get into it, but it's the first of the 3 attempts that scientists use before CRISPR and CRISPR-Cas9 was even discovered. So it works by adding genomes into body cells of a person in order to try and treat a condition or a disease that the person has already developed. So it's treating something that a person already has. So obviously the person's born grown up, you know what I'm saying, and they have the condition, and it's trying to, gene therapy is the prospect of trying to treat or eliminate that condition that the person has already developed. And does that make sense, Jordy?
Yeah, I think so. But quick question.
Yeah.
Here. So, which is there a more or a better therapy than the others?
I mean, they're different. They're all different types. They're all used for very different things. I'm going to kind of explain what each one is, and you'll kind of understand why they're so different from each other. Okay. You'll kind of see a little bit more of why they kind of have to get broken up into these subcategories, because they're so different from each other. You can't really kind of compare each one to each other because they're so different. So I'll kind of explain that as I get into the other 2 types.
I have one more question.
Yeah, no, I got you for sure. Yeah.
When you say they add genomes into the body cells, so they don't really do the nucleotides with the gene therapy one. It's different than CRISPR.
Well, yeah. So gene therapy doesn't even use CRISPR or
okay.
CRISPR-Cas9 technology. It's just it's using different inserting different genes to try and treat or replace. So I'll give an example so you can kind of understand a little bit further. So a good example that I found was actually a pretty cool experiment that a company did with people with a disease called Leber congenital amaurosis, if I pronounce that correctly. It's a disease in which genetic defects in the retina's light-sensing photoreceptor cells cause patients to lose their sight over the course of their childhood. And
That's terrible.
Yeah, it's really sad, actually. So I know that's kind of wordy, so I'll kind of explain in more simpler terms. But basically, this genetic disease blinds you over time by the deterioration of cells inside of your retina, so inside of the light-sensitive receptors in the back of your eye. They're just deteriorating over time throughout your childhood, and eventually you go blind from it. So using gene therapy in 2017, scientists from Editas Medicine injected a harmless virus. So they injected a virus through eye drops over the course of about 6 months to deliver a working copy of the RPE65 gene into those retina cells in the back of the eye to try and replace and regrow that gene so that they could see again over time. And it was actually extremely successful. I think they experimented on 6 people, and 3 people found successful results. So that's a 50% success rate, and for something that's so new and so risky, that's profound success. And actually, they even recorded the results from one of their patients. I don't know the name, obviously, but it was a 54-year-old woman who was completely blind from Leber congenital amaurosis, and she injected the RPE65 gene into her eyes for about 6 months, and after the 6 months was able to depict almost every shape that was in her sight, almost like shadows, just without the fine details and the color, unfortunately. But compared to complete blindness before that, that's it's pretty amazing that she was able to regenerate those cells, so she was able to somewhat see again.
Yeah.
So that's kind of what gene therapy is. It's the least complicated of the 3, and it doesn't even really use CRISPR technology. But without gene therapy itself, then the rest of the categories of genetic alteration wouldn't even be a thing. And so I'm going to kind of get into this second one, which is hereditable genome editing. And it's far and away the newest category of genetic alteration, just because it's spreading rapidly so recently, and it's definitely complicated and scientific. So I'm going to kind of explain how it works, and then I'm going to kind of try and simplify it in a way that everybody can understand. So it works by designing a CRISPR RNA, so a guide RNA that can target a specific gene or a sequence of DNA. So kind of like the dotted line on the paper that I was talking about. And then it introduces the guide RNA, so the CRISPR, to the Cas9 protein, which are the scissors that I was talking about earlier. And they do that to answer your question from earlier, to an early stage embryo. So before the embryo has developed. Yeah, exactly. So it's an embryo. And they introduce that CRISPR-Cas9 combo to the embryo, and then that tandem cuts the DNA at the targeted location that the scientists are looking at, and then that embryo's natural repair mechanisms then try to fix the DNA by deleting or replacing the targeted sequence. with the desired genetic sequence, and in turn, it gets rid of the DNA sequence that was cut. So it basically just completely eliminates the DNA sequence that was, potentially harmful or potentially was causing a genetic disease, or was going to cause a genetic disease to the embryo. So I know that's kind of, complicated, but did that, somewhat make sense?
So, after the CRISPR sequence works, then they cut out the part of the DNA. What happens next? Do they just add in another?
Well, yeah. So naturally, the embryo in humans, they have a natural repair mechanism.
Oh, okay.
And they fix the DNA by just replacing it. So they'll cut the targeted sequence of DNA, and then it'll just immediately get replaced. Because that's kind of just how it works.
By CRISPR or like body?
Naturally. Yeah, naturally. So CRISPR is just guiding. It's a RNA molecule that's just guiding the Cas9, and then the Cas9 is what's cutting it. So the Cas9 is like the scissors, and it's cutting that sequence of DNA, and then that DNA just gets eliminated and replaced with the healthy sequence of DNA. And we'll kind of get into. I mean, in this case scenario, you're just replacing the cut sequence of DNA with just a healthy sequence of DNA, but potentially in the future, scientists are trying to figure out how to replace the cut sequence of DNA with maybe an advanced sequence of DNA, and that's kind of the third subcategory of genetic alteration. So I'm gonna get into that here in a second. But this process is way different from gene therapy because you're actually altering the sperm and the egg, so the gametes in the embryonic stage to prevent the disease or disability from existing altogether, instead of trying to treat it in a person, that's already has it. So that's why it's so different from genetic therapy. And you were asking how to compare them, but it's really not possible to compare them because they're two completely different things. You see what I'm saying?
So this is like the newest technology.
Yeah, this is like what's currently, kind of being discovered almost. And it wasn't possible before CRISPR-Cas9, was discovered. Unlike gene therapy, gene therapy's been happening for
years,
over 50 years. Yeah, and but this is just something that's kind of just now coming up in the science world. So it's a absolutely revolutionary discovery. And I'm kind of gonna explain what it means for science in the future of science later on. But I just wanted to kind of go over what it is, so we understand how it can change things. And I'll kind of segue into the third subcategory, which is genetic enhancements. And this one's pretty simple to understand, and it's pretty cool, actually. It's probably the coolest of the three, although it's extremely scary. And it's basically the same thing as heritable genome editing, except it's where a non-therapeutic alteration of genes aimed for improving cognitive and physical functioning that was already at or above the normal level for a person. So instead of targeting a sequence of DNA that has a harmful genetic disease, it's just targeting a regular segment of or sequence of DNA and replacing it with an advanced sequence of DNA. So, I mean, do you understand kind of what I'm saying?
So, you basically are modifying to enhance?
Yeah, so, so kind of like an example would be like, you are cutting a sequence of DNA and genetically editing an embryo to give it the genes needed to have, let's say, twice as much muscle growth as a regular human organism, or I mean, this is kind of more of like a far-fetched example, but
Like hair color, maybe.
yeah, like even hair color, or like it even goes as far as splicing DNA from a type of dog, um, and cutting it and replacing a sequence of human DNA with that dog DNA, so that you enhance the human scent
Wow,
abilities.
that's
So
really
that's
crazy.
yeah. So that's just like a basic. That's really a far fetched example, but it's something that's um, is crazy, but it's a realistic possibility of genetic enhancement. So um, you're basically talking about superpowers here,
Yeah,
which is pretty cool, but it's also extremely terrifying, and there's so much that kind of goes along with it. Um, and it sounds kind of like it's straight out of a movie, um, but it's realistic, which is kind of crazy to even
hmm
think about. And it's far and away the most open ended type of genetic alteration. So it's really like it's newer than heritable genome editing, and it's barely introduced, and there's a lot of like regulations that some countries have against it, um, and they've like implemented regulations so that it can't even become a thought in people's heads. But eventually, there's going to be a time where those regulations, aren't really there anymore, and that's just a frightening thought, because then, certain people are just splicing DNA with anything that you can even think of to advance human performance. Yeah.
yeah. Quick question. So while we're talking about genetic enhancements, do you think that this is considered like playing God as a
Oh,
controversy?
absolutely, absolutely. And I'm kind of going to get into that when I talk about the ethics um, of genetic alteration. But yeah, I absolutely think it's somewhat playing God. And there's actually like there's a lot um, of worry in the scientific community because of that. There's not really a technology out there right now that's as powerful as genetic alteration, and essentially it could be changing human life as we know it. So it's completely playing God to a certain extent, which is why it's so frowned upon. um, Genetic alteration is because of the worry of genetic enhancement and how it could negatively change the world as we know it. So um, yeah, I mean it's definitely playing God to a certain extent, for sure. Um, but with with the um, with your newfound knowledge of genetic alteration and the three different categories, with that being therapy, HG, so heritable genome editing, genetic enhancements. I hope that you understand a little bit more the potential that genetic alteration has to absolutely revolutionize medicine and human health as we know it. But it also raises major ethical concerns, like you were talking about, playing God, that has to be addressed. And the history and development of molecular genetics has been haunted by the specter of kind of playing God. Um, and so I kind of want to get into that a little bit, and the negative effects that genetic alteration can cause on the world as we know it. So
So going back to I think the CRISPR
uh,
and the Cas9, a question I have for you is, um, are there any altercations or progressive abnormalities after CRISPR is performed, or has there been any cases that you've known of that maybe they took out?
like negative effects
Not
of
necessarily,
it.
like maybe they tried to take out, you said, what disease did you say?
It was
Polycystic?
yeah. Well, I mean, it's a disease that makes you blind over time.
Well,
is that what you were talking about?
Yeah. Does that ever come back?
So that one, it just happened in 2017.
Okay.
And I don't believe that that 54-year-old lady that was able to see again, was reblinded. But that's not even. I mean, that's genetic therapy. That's not even CRISPR or Cas9 playing a role with the healing of that. That's just genes being replaced and healed over time. But CRISPR-Cas9 is essentially just eliminating even the potential for that disease to develop in children for good. So it's kind of cutting out that middleman and just eliminating the disease altogether. So yeah.
All right.
Yeah.
One more question. We didn't really answer it yet, but how successful are all these three, or say surgeries?
I mean, yeah. I mean,
has there been
essentially,
any cases?
I mean, it's kind of. I mean, it's not really surgery because it's before the embryo is even developed, but there's been multiple successful attempts at genetic alteration. There was a scientist in China that did it without permission from the government on a set of twins to completely eliminate the chance of them getting AIDS altogether. So that
Weird.
Yeah, in and of itself. I mean, they obviously. I don't think maybe they would have gotten AIDS, maybe not. As adults, but he eliminated even the chance of it by cutting that segment of DNA just out of them completely, and that's crazy to think of because he cut that segment of DNA out. Now, future generations that are birthed from that set of twins will also have that segment of DNA cut out as well. So it's not even possible for future generations of that from that set of twins to even have AIDS altogether, which is absolutely crazy to think about. As I was saying earlier, with this this technology being so powerful, there's a lot of ethical concerns of it. One being eugenics, which is essentially the thought of perfection in human form. This is something that was brought up during World War II. It was something that Hitler was chasing, the thought of eugenics in a human being, and he was trying to create it. Obviously not using CRISPR-Cas9 technology because that was not even a thing back then, but that's essentially what it means. And that's a big fear in the genetic alteration community because it could really, really, really easily lead down the slippery slope of genetic enhancement for eugenics. So that's a really scary thought. But essentially, I just wanted to kind of give you a thought in your head of what the world could look like if that was to take place. So I want you to close your eyes, Jordy, and everybody listening, and I want you to imagine that you are in the year 2075. So I know that's far down the road.
Flying cars.
Yeah, but just hang in there. I'm gonna explain. So imagine you're in the year 2075, and I want you to imagine that you're a newlywed with the love of your life. You know what I'm saying? You guys just got married, and you're making the decision to have a child. Unfortunately, in 2075, this is no longer done the old-fashioned way. So you go to the childbirth lab in the city, and you and your spouse take your whatever futuristic car, whatever whatever the transportation is in the future. You take that to the childbirth lab in the city. On the big day, you decide to have your, or I guess, plant your embryo, and you enter the building, and soon after, a lab worker introduces you to the menu booklet, and you begin designing your first child. So this is kind of a
so it's like Build-A-Bear.
Yeah, it's literally like Build-A-Bear. It literally is. So this is kind of where the term designer babies comes from. It's a popular term in the genetic alteration community because this is a realistic possibility of what could happen in the future. So I'm going to keep going. Keep your eyes closed. So imagine you and your spouse, you're at this childbirth lab. You have this booklet in front of you, and it has all these different topics, all these different things you can get for your kid. But let's say you both agree to get the basic safety package that includes immunity from all the genetic diseases that could fall into the child. And you also agree to pay a little extra money to get the no birth defect package for your child as well. Because that's something that, costs more money, but it's something that most people do with their kids, obviously, because you don't want your kids to have birth defects, right?
Yeah.
So your spouse wants a child. Let's say your spouse wants a child to be an athlete. So they buy the athlete package for your kid, which includes 6 foot plus height, big hands, fast metabolism, and a 1.5 times advancement in muscle growth. This is, it sounds crazy to even think about, but this is like, this is somewhat possible, right?
This is possible. That's so crazy.
Yeah, and let's say you personally decide you want your kid to look like a model. So you drop a buttload of cash on the advanced looks package that includes a sharp jawline, proportional face, profound cheekbones, and a genetic guarantee of limited acne as a teenager.
And blue eyes.
Yeah, and sure, blue eyes. And once you just finish deciding all these specific parameters that you want for your kid, you hand in your menu booklet, you go inside the lab, you extract samples of the gametes, so the sperm and the egg, and from then you are given the price that it will cost, which will most likely be some absolutely outlandish, outrageous price, and you pay, and then you schedule your next appointment, which is inserting the genetically modified embryo into either you or your spouse through in vitro fertilization. And from then, the pregnancy begins with this super baby, basically, this designer baby. So, it sounds absolutely ridiculous, but it kind of gets you to paint a picture in your head of what the future of genetic therapy looks like as it begins to get discovered and it begins to be successful. So, what do you think about that? What, as I kind of explained that, what were the thoughts going through your head?
Yeah, that's definitely crazy to just build your kid just like how you want it. It's like Mr. Potato Head.
Yeah, sure. Just like Mr. Potato Head. Yeah, that's an absolutely insane thought.
You can put the ear in the mouth, do whatever you want. But you're building it how you want it to be like.
That's exactly.
The same way you're building your kid.
Which is just not even it doesn't even register in my head. It doesn't even. But
But there's gotta be it has to be playing God and so much in that it's just not ethical.
It's so unethical, and which is why it's such a controversial topic to discuss because it could be so good for eliminating genetic diseases, all genetic diseases, but it can be so bad too, in a sense of that potentially happening and that being our future. But I wanted to highlight that when talking about ethics and eugenics and science, they're deeply entwined within each other. Science and ethics are, because the work of science is creating new ethical issues, and the response to these feeding back to what influences what sciences can or cannot do, or researchers can or cannot do. So like ethical concerns can potentially stop a specific scientists from continuing research on specific things because of the thought of what I just went over, like of what the future could look like with certain things like that. But when it comes to altering the genetic code of organisms, there's still so much that we don't know. And CRISPR-Cas9 has helped us learn more than we have ever thought we could achieve within genetic alteration. But we're just now learning what could happen. You know what I mean? And we really don't even know the extent of it yet. And scientists are still trying to fully understand how genes interact with each other. And there's always the risk, of eugenics and ethical concerns that we can't even begin to think of. The room for evil is so large. But the crazy part is that it doesn't even make sense to outlaw something like it. Like when you think Build-A-Bear, for your kid, designer babies, you think instantly, this needs to be outlawed, right? Like this needs to be illegal. Yeah. We have to stop this immediately, right? Like that's something that you think, even if it's potentially healing genetic diseases, it's just not something that any human should be messing around with because it's just playing God. And that's what most people think. But the crazy part is that if you were to outlaw something like CRISPR, since it's a bacteria, it's actually accessible to anyone. So like you and me could get a kit for CRISPR if we really wanted to.
Oh, so anybody can.
Yeah, so because it's a bacteria, it's found in the body.
Gotcha.
So it's something that anybody can purchase, and there's people that sell kits, CRISPR kits online. And so to an extent, it doesn't make sense to outlaw something like that, because if you're outlawing it, then you're causing scientists to halt research, immediately, right? Yeah. Completely stop whatever research they're doing with genetic editing. But there's still those kind of rogue scientists in their garages, mastering CRISPR, while the actual researchers can't do anything. You know what I mean? So it just doesn't make sense to make something like that illegal, because you're not stopping anything. Yeah. You know what I mean? And essentially you're just, you're opening the door to genetic enhancement in the worst way possible. So that's kind of a crazy thing to think about, is that it can't even get outlawed because it's already too late. You know what I mean?
Yep, for sure.
And so I mean there's people out there, and scientists out there, in their own garages, in their own homes, that are already accomplishing some wild feats, actually, if you didn't know, such as working towards producing glow-in-the-dark dogs. Wow. By combining a glow-in-the-dark fish.
Well, I guess you'll never lose them.
Yeah, they're segments of DNA from a glow-in-the-dark fish with a dog to try and get glow-in-the-dark dogs. And that guy's doing that in his garage with a CRISPR kit. And then there's people trying to give themselves through gene therapy and gene editing, because obviously they're already born, the ability to stretch further than the ordinary human. So Mrs. Incredibles type stuff. Yeah. Through CRISPR technology. And so, just thinking about that, it's kind of crazy, because it's not something that you can stop anymore, because it's already so far. Yeah. But it's really scary to honestly think about something like that. So, kind of to sum up the ethical and the prospect of eugenics, and kind of that portion of genetic alteration, you kind of have to think of the future and the potential for genetic enhancements, and how bad that could possibly become. So that's kind of why I told that make-believe story, because it kind of puts into perspective how realistic something like that becomes with genetic enhancements. And if something like that happens, it could lead to a very, very vast genetic divide between the rich and the poor, meaning that wealthy individuals or countries with the resources to fund gene editing technologies could potentially create a genetically superior class of humans, while those who cannot afford it would be left behind. So it'd be just widening the gap between the high class tier of humans and the low class tier of humans. So you think genetic editing is like eliminating these genetic diseases and all this really good stuff, but to an extent it could widen the gap of what it already is between high class humans and low class humans. And some argue that altering the genetic code of an organism just violates the sanctity of life itself. But obviously others argue that it's just simply a form of intervention that can lead to better quality of life through curing genetic diseases. So it's really a controversial topic, but it's something that not a lot of people know about. So I wanted to inform kind of what it is, how it works, what the future looks like, the ethical concerns behind it, and something that I just felt like more people need to know about. And so while genetic alteration and heritable genome editing hold great promise for the treatment of genetic disorders and the improvement of human health, they also raise like really, really significant ethical concerns that we have to approach cautiously about. I think together the world has to consider the ethical implications, ensure that we use them in a way that benefits society as a whole, instead of trying to push towards advancing one area and leaving the other area in the dust, because that's just tearing the world apart even more than it already is. And it's really honestly disappointing that it's so unknown today, because it's such a profound topic, and honestly it could be the most important discovery and piece of technology in the last 100 years, because it can completely change the face of human life as we know it. And so I hope throughout this podcast I provided all of you guys with an informative walkthrough of genetic alteration with CRISPR and Cas9 technology. And I hope Jordy, you and others listening are able to inform others of the prospect of gene editing now as well.
Yep, for sure. Will do.
I appreciate you for taking the time to sit down and talk with me about it. And I hope everybody learned something new. So with that being said, that's all.